Okay, so this is the last lecture for the POD pathology. Okay, and let us discuss some basics here. I'm going to discuss about the cell cycle. Normally, the quiescent stable in a stable tissue, the cells are in resting stage. Okay, so in a stable tissue like liver, pancreas, kidney, the cells will be in a resting stage. So this is a cell. So this cell is from the stable tissue. Okay, the cells in the stable tissue we already discussed in the tissue repair chapter. They are called quiescent cells. They are called quiescent cells. So they are called quiescent cells, and the quiescent cells will be in a resting stage. Normally it will be in the resting stage. For example, they are the resting stage in cell cycle is called G0, growth 0. Okay, and why the cells are in resting stage? Because of certain genes inside. They are called growth inhibiting gene. They are called growth inhibiting gene. Okay, so otherwise called as tumor suppressor gene. Tumor suppressor gene. Okay, so why the cells are in quiescent stage, why the cells are in non-dividing stage, why the cells are in G0 stage, okay, in a stable tissue? Because of these genes are called growth inhibiting gene. Okay, until this gene is active, they will not allow the cell to go into division, they will not allow the cell to go progress in the cell cycle. So up to when this gene can be active? So if there is any stimulus, for example growth factor, if the growth factor is stimulating a cell, that will make this growth inhibiting gene to off. They switch off this growth inhibiting gene. So the growth factor which stimulates the cell, that switch off this gene, and they activate the another set of genes. They activate the another set of gene. They are called growth promoting gene. They are called growth promoting gene. So once this gene gets activated, normally this gene never get activates. If they are stimulated by the growth factors, this gene will get activated. Then they will push the cell from G0 to G1. They push the cell from G0 to G1. So the cell is entered the cell cycle. So what is the outcome of the cell cycle? They have to divide and they form two daughter cells, right? That is the outcome. So for example, when we are teaching to the POD students, what is the outcome we expect? We all pass in a good score. So that is the expectation of any teacher. Likewise, what is the outcome of this cell cycle? Is they have to divide into two cells. They have to make two daughter cells. So for that, first thing they have to do is they have to synthesize the DNA. So from the once they enter the cell cycle G1, they go to the stage called S stage, that is synthesis of the DNA. They enter into the next stage called S stage. So synthesis of DNA is very important event, because the DNA is the one which determines the fate of the cell. So the DNA is very important. So when they synthesize the DNA should be synthesized normal. Okay, so DNA should be synthesized normal. So somebody has to check the DNA whether it is synthesized normal or not. For example, I know the PhD students all study good, that doesn't make any sense. It has to be checked by the exam. So likewise we have to check, because I am going to talk 12 o'clock about the exam. So that's why I am putting examples of PhD exam. So now somebody has to check it, right? So when somebody has to check it, that area is called checkpoint. That area is called checkpoint, and this is called checkpoint 1. This is called checkpoint 1. This is checkpoint checkpoint 1. Okay, so this is very important checkpoint, because here there are some genes going to check the DNA whether it is synthesized correct or wrong. Okay, so these genes generally we call it as growth regulating genes. What are they? Growth regulating gene. Yeah, growth regulating gene. So in the growth regulating gene we have two genes. Okay, one, while they are checking the DNA, if the DNA is having some error, DNA is having some problem, they will repair the DNA. They will repair the DNA. They are called DNA repair genes. They are called DNA repair gene. Okay, if they can't repair the DNA, okay, they cannot able, they try and they are not able to repair the DNA. So immediately another gene comes to a role. That gene is called apoptic gene. So they tell that that cell, okay, I cannot able to repair your DNA. So if you come out, you will come as a bad cell. So you have to die. So that cell will go for cell death. So that will be done by a group of genes called apoptic gene. That is called apoptic genes. Understand? So these are the growth regulators. So they check the DNA at checkpoint 1. Okay, now they checked the DNA and DNA synthesized is good. Then they will progress to the next stage called G2. Once they reach the G2, then they go to mitosis. So then once they go to mitosis, there is the end stage. They are going to divide. They are going to divide. So when they go to the mitosis, then that is the final checkpoint. So there is a checkpoint between the G2 and M. This checkpoint is called checkpoint 2. Checkpoint 2. Again the regulatory genes will come and check everything is correct. If it is correct, they will them to go for division. So they divide into two cells. And one will come for the G0 stage and one will enter the G1. Okay, so one cell is preserved. One cell is preserved. So likewise the cell cycle continues. So when the cell is at resting stage, it is called G0. The cell is at the resting stage because of the genes called growth inhibiting genes. If they are stimulated by the growth factors or any other stimulation, the cell moves from G0 to G1 because of two reasons. One is growth inhibited genes are switched off and the new genes are activated, growth promoting genes are activated that push the cell from G0 to G1. Once it enters the G1, then they go to synthesis of DNA. So this is an important event, so they check that event. So there are growth regulatory genes that check the DNA. If the DNA is having some error, they will repair the DNA. If they cannot able to repair the DNA, then they will induce the apoptosis, that is apoptic genes. then they will move the cell to S to G2, then the G2 to M. So in between the G2 to M, they again check the cell, and if it is good, they will allow them to go for division. So they will form a the final two daughter cells. So this is the normal cell cycle. Okay. Now, if there is any problem in these four groups of gene, one is growth inhibitory gene, growth promoting gene, DNA repair gene, apoptic gene. If there is any problem in these four genes, four groups of genes, then it results in cancer. It results in cancer. I will tell you one example. If there is problem in the growth inhibitory gene, okay, there is a problem, the growth inhibitory gene is not active, what will happen? The cell will not be in resting stage itself. The resting stage is completely absent. So the cell will be always in the cell cycle. The cell will be always in the cell cycle. Okay, if there is problem in the growth promoting gene, that is the growth promoting gene is in always active without stimulus. Without stimulus, the growth promoting gene is active. What will happen? So there is no again G0 stage, which means the cell is always in the cell cycle. The cell is always pushed to the cell cycle. Okay, if there is DNA repair gene is absent, they cannot repair the DNA. So abnormal DNA is formed, that will be not checked and they will come out. Okay, and if there is no apoptic gene, what happens? The cell will not die. So they will live long and they will divide more. So these are the problems happens in the cancer. So this is about normal cell cycle. So with this basics, let us go to the slides.